Vitamins and minerals, sodium. We will now discuss sodium and its role in electrolyte and fluid balance. We can see here sources of sodium. So food sources of sodium would largely be processed foods, as food processing and manufacturing often requires sodium. We also see that it is naturally occurring in foods such as milk, meat, eggs, and vegetables, but this is largely what will be in lower quantities than in more processed or manufactured foods. We have different categories referring to different types of foods as being free of sodium, very low in sodium, or reduced or light as it relates to sodium. And we'll define each of these. So sodium free would be a product that contains less than 5 milligrams of sodium per serving, so that'd be less than 5. Very low sodium would be less than 35 milligrams of sodium per serving. Low sodium would be less than 140 milligrams per serving, and reduced or light sodium products would be those products containing less than 25% of the sodium compared to the original product. So we can see that in general in the United States, sodium intake is largely not matched to sodium needs, so intake will actually be significantly higher than what is needed. We see that according to the Institute of Medicine, the daily recommended sodium intake is less than 1,500 milligrams per day, and this is an adequate intake level. However, we see that average daily intakes in the US is much higher than the 1,500 milligrams per day. So in adult males, it's approximately 4,200 milligrams per day, and in females, 3,300 milligrams per day. Sodium excretion, so we'll talk a little bit about this process. It's regulated to maintain proper osmolarity in the extracellular fluid. So typically we have large amounts of sodium excreted in urine. We see that intakes are often higher than what is needed, so excess amounts would need to be excreted in urine. We also have some excretion of sodium in sweat and feces, but under normal conditions the amount excreted through these means would be relatively low. Some individuals are known as salty sweaters, so they excrete more salt through sweat, but this would not be the typical individual. We'll look now at the process of absorption. So the process of sodium absorption, there's actually 3 different modes that we're going to review. Since there are a number of mechanisms for sodium absorption, we can see that the percent that is absorbed is very high. So it is approaching 100% absorption of that which we consume. So we'll go over the 3 pathways, so one being a sodium glucose co-transport system, so sodium and glucose being absorbed together. And then we have the electroneutral sodium and chloride co-transport system, so sodium and chloride having a concurrent absorption mechanism. And then lastly, we see this electrogenic sodium absorption mechanism. So we're going to review each of these 3 pathways of absorption. So first we see the sodium glucose transport system. So we can see here in a kind of more general and simple terms, we see that as glucose enters the enterocyte through the brush border membrane, that sodium follows. So we have sodium and glucose concurrently being absorbed into the enterocyte through the brush border membrane, and then for sodium to be transported out through the basolateral membrane, we have activity of the sodium-potassium ATPase. We'll talk about this ATPase in more detail. However, essentially it is pumping sodium out of the cell while bringing potassium into the cell. So this sodium-glucose co-transport will be the first mode of absorption. Next, we have this electroneutral sodium and chloride absorption. So we see here concurrently sodium entering the cell and a hydrogen ion being pumped out of the cell. And so at the same time as sodium entering the cell, we see chloride entering the cell, and this is going to actually balance the charge with the sodium entering, we see the chloride entering, and then we see bicarbonate that will be exiting as chloride comes into the cell, then the bicarbonate will be going out of the cell. And then at the same time, through the basolateral membrane, we see sodium exiting through the sodium-potassium ATPase. So sodium exits and then potassium will come into the cell, and then we also see chloride leaving the cell. So we have this electroneutral process with both sodium and chloride entering the cell and exiting through the basolateral membrane. Lastly, we will see the electrogenic sodium absorption process. So in this process, we have a channel, a sodium channel, where sodium will enter through the brush border membrane. And then very simply, we see that it will then exit via the sodium-potassium ATPase. So just to talk a little bit about the transport and the functions of sodium. So sodium is transported freely in blood. So it will be transported in the free form through circulation. And we see some of its main mechanisms here. So we see its involvement in the maintenance of fluid balance. So we'll talk about this process and the maintenance of fluid balance specifically in more detail. We'll talk about its involvement in the renin, angiotensin, aldosterone system. So we'll see how we have the involvement of sodium and the renin, angiotensin, aldosterone system. This system also regulates the levels of the blood, so blood volume, as well as blood pressure. Also, we're going to see the role of sodium in nerve transmission and impulse conduction, so nerve conduction and muscle contraction. Okay, so we're going to see here the electrolyte composition of body fluids. So we see a number of electrolytes. So we see cations here, we see anions, and other components such as protein that will be existing in plasma and also within the cells. So we can compare the levels of these electrolytes, the cations and anions, specifically within plasma and then within the cell. And so just to take a look at those that are going to be existing at higher concentrations within plasma and those that will be existing in higher concentrations within the cell. So as it relates to plasma, we see specifically that sodium is going to be in higher concentrations in plasma. So it will be in higher concentrations outside the cell versus inside the cell. So higher concentrations of sodium outside the cell, and this creates a sodium gradient. So with higher concentrations out of the cell, lower concentrations in the cell. And this is achieved through the activity of the sodium-potassium pumps, or the sodium-potassium ATPases that we saw involved with the process of sodium absorption. We have these sodium-potassium ATPases on numerous cells in the body, and so the sodium-potassium ATPases create this gradient, and with this gradient, actually, we will facilitate the process of nerve conduction and muscle contraction. So we're going to talk about how this sodium gradient actually assists with the depolarization of cells and promotion of action potentials. On the other hand, we see how potassium is opposite, where it has lower concentration outside the cell and higher concentration inside the cell. Other electrolytes where we see higher concentration outside the cell is chloride. So like sodium, chloride is also at higher concentrations outside versus inside the cell. Like potassium, magnesium has higher levels inside the cell versus outside the cell. We'll now review here in more detail the processes and functions of the sodium-potassium pump, also known as the sodium-potassium ATPase. So we can see here in this diagram, the sodium is in blue, right? So we have these sodium ions in blue and the potassium ions in orange. We also see involvement of ATP, and ATP then being hydrolyzed and then powering the activity of these sodium-potassium ATPases. So we first see here that this is going to be inside the cell, and we have here being outside the cell. So the purpose of these sodium-potassium ATPases is to take sodium out of the cell and to bring potassium into the cell. So for the first step, we see the binding of 3 sodium ions from inside the cell to the sodium-potassium ATPase. Also, we see binding of ATP. Next, we have the hydrolysis of ATP, and the phosphate group then binds to the sodium-potassium pump, the sodium-potassium ATPase, and at that same time, the sodium will exit the cell and go into the plasma fluid. So we have then sodium going from inside the cell to being released outside the cell through the sodium-potassium ATPase. Then we have potassium from outside the cell binding to the sodium-potassium ATPase, and then we see with release of the phosphate group, then that powers the entry of the potassium inside the cell. So essentially we have the sodium going from inside the cell to outside the cell, so 3 sodium ions, and then 2 potassium ions going from outside to inside the cell. So the activity of these sodium-potassium ATPases are going to be very important in creating this gradient of more sodium outside the cell and more potassium inside the cell. So you can see in this diagram, we're going to see how the concentration gradient of sodium, how it's developed by the sodium-potassium pumps, will play a role in nerve conduction and muscle contraction. So specifically here, we see this axon terminal, we see this neuromuscular junction, we have a neuron, and we have a muscle fiber. And we can see the nerve impulse from the neuron here will be then transmitted to the muscle fiber, promoting a action potential and then a muscle contraction. So sodium is going to play a significant role in this process, and sodium's ability to rush into the cell as these sodium channels open, the ability of sodium to rush into the cell and then promote a depolarization and thus an action potential, is going to play a significant role in the stimulus for promoting muscle contraction. And the reason that sodium can rush into the cell is because of the concentration gradient, with higher sodium outside the cell, lower sodium inside the cell, so it wants to go from high to low concentration. So just a little bit about this process, and we have seen this in previous slides, especially when discussing calcium and the role of calcium in this process. But we see that here as these vesicles containing acetylcholine release acetylcholine in this area of the motor end plate, we see that the acetylcholine binds to the acetylcholine receptors on these ion channels. And as the acetylcholine in these little round circles in orange, so as this acetylcholine binds to the acetylcholine receptors on these ion channels, the ion channels open and sodium rushes into the cell. And it again rushes into the cell because it is at a higher concentration outside versus inside the cell, so it rushes into the cell. This also promotes these voltage-gated sodium channels to open, further allowing sodium to rush into the cell. And so the entry of high concentrations of sodium rushing into the cell promotes a depolarization and an action potential, which then would promote muscle contraction.